Hello guys, welcome to the Knowledge Room, the concepts of life sciences and biotechnology. Today we are going to learn the anatomy of gene, or we delineate the different parts of a gene. So before going into that, we all know that the information is stored in the genome or in the DNA of the nucleus of a cell. So whatever information is there, this information will be transcribed to prepare the pre-mRNA. Then there is a processing steps by which the mRNA will be prepared, and this mRNA will move from the nucleus into the cytoplasm, where it will take the help of the ribosomes and the other component to prepare the protein. Finally, which is doing all the job. So this is the central dogma in which the information that is stored in the DNA, that is transferring its message from the DNA into the protein so that the job will be done. So the gene, that is the information. By the classical genetics, they are the bits on a string, or the DNA is the string. For the molecular geneticist, the same gene is denoted the string on the bits, considering the nucleosome. Because the nucleosome in the chromosome packing, it looks like a beads, and the information is present in the DNA that is wrapping this nucleosome, or the histone that is forming the nucleosome. So this is the string that has the information. So according to the molecular geneticist, the genes are the string on the beads, that is the nucleosome. But according to the classical genetics, genes are the small portions of the locus in the chromosome that are responsible for each trait. Next is when you go to the chromosome, the heterochromatin or dark stained chromosome are tightly packed, and the euchromatin are the loosely packed, they are transcriptionally active. Then eukaryotic genes, unlike the prokaryotic genes, they are not collinear with their peptide products. So that we are going to discuss. So this is the beta globin that is responsible for carrying oxygen in the RBC. So we are understanding the anatomy of a gene, or we are studying the different parts of a gene by taking example of the beta globin. So this area is one gene. So in this one, we can see in the extreme left, we see the promoter. So what is the promoter? Here we can see there is a RNA polymerase II binding, because it is involved in the transcription. So to execute the transcription process, there is the need of the RNA polymerase II. So promoter is the region of the DNA where the enzyme that is responsible for the transcription of the RNA synthesis. One of the examples is the RNA polymerase II. It initiates the transcription, it binds in the promoter area. So it has the three distinct units the promoter has, that extend several base pairs before the transcription initiation site. So this area is the transcription initiation site, and we can see this promoter is present upstream from the transcription initiation sites. And in this promoter, we can see there is a TATA box, or the TATA binding protein. So this is the sum of the promoter, they contain the TATA box. What is the sequence of that? It is the TATA. Next portion is the transcription initiation site. So in this gene, this is the transcription initiation site. Where the transcription initiate, that is responsible for the preparation of the mRNA. This is also called the cap sequence. It calls for the addition of a modified nucleotide, the cap. So we see this is the synthesized mRNA. and we can see at the 5' end there is a cap, G triple PAC. So this transcription initiation site, this is also called the cap sequence. So immediately after the synthesis of the mRNA, it is capping it at the 5' end after transcription. And the cap sequence begins the fast exons. So this is a fast exon. So what is an exon? The coding sequence that also we'll discuss here. So after the transcription initiation site, we see a region that is the leader sequence, this yellow area. So what is this leader sequence? We can see there is a translation initiation site and there is a transcription initiation site. So the area of the genomic DNA or the DNA between the transcription initiation site and the translation initiation site, this is called the 5' untranslated region. It is untranslated means this is not the part of the polypeptide chain after the translation, and this is also called the leader sequence. So as I explained, this is the intervening area between the initiation point of transcription and translation. And what is the role of this leader sequence? It determines the rate at which the translations will be initiated. So till now we have discussed three areas of the gene. The first one is the promoter, where the RNA polymerase II will bind. Second was the transcription initiation site, that is also the cap region. And the third one is the leader sequence. Next one is the translation initiation site, means that this is the first amino acid, that is the first amino acid of the polypeptide chain that is translated. So this is usually the ATG, and in the recent discoveries there are some exceptions. So the ATG, that is a part of the DNA, it will become AUG in case of the mRNA. But whenever we see the genomic DNA, the sequence, the ATG is the start codon. Usually the codon it is given to the triplet nucleotide bases of the messenger RNA, but most of the time the people they usually use the terminology codon for the genomic DNA sequence also. But technically this is not right. Same in every gene except few exceptions, and distance from the transcription initiation site varies among different genes. So there is a transcription initiation sites, and where the fast translation, the translations will be initiated by the fast amino acid of the polypeptide chain. This distance it varies from the gene to gene. Next is the protein coding region. As I told you, the exons. These are the protein coding regions. So the number of the exons is varies depending on the gene, the species, and the same gene in the different species. The exons number of the exons it varies. So exons, another terminology is the protein coding sequences. And the area between the protein coding sequences, the non-coding areas, those are called the introns. So the interspersed with the non-coding sequence of the introns, there are the different number of the exons and the introns depending on the gene. Just now I discussed that one. So next one is the translation termination codon. So this is the gene that is first transcribed at the pre-messenger RNA, then there is the messenger RNA, and the messenger RNA, it will use all the translation machinery in the ribosomes and it will prepare the polypeptide chain. So where the translations will be terminated, it is decided by the codons. In the DNA, there are synonyms are TAA, TAG, and the TGA. So this is the translation termination codon. TAA. This red area is the translation termination codon. And when a ribosome encounters during the translation process, in this process, when this is encountered this area, this is the same. The after the transcription and the processing, then the translations will be terminated and the protein is released after this one. So after the translation termination codons, we have the 3' untranslated region. This is the 3 days on translated region. After this, like that, we have the 5 days on translated region here. This is the cat, right? 5 days on translated region. Like that, there is the three days on translated region that include the sequence of double A, T, double A in the repeating one. And this is also called the polyadenylation. So we can see that this is after the transcription, there is a poly T. So this is the polyaddition site. This is the 3 days on translated region. So it almost constitutes 300 to 300 repeating adenylate residues like this one, and there will be T in between in the RNA transcript. What is the role of this 3' UTR? It confers stability on the mRNA, allows the mRNA to exit the nucleus, permit the mRNA to be translated into the proteins. Then after the 3' UTR, we have the transcription termination sequence. This one is the transcription termination sequence that continues beyond the polytail, and it is about 1,000 base pairs before being terminated. So a gene that at the various regions that starts with the promoter in summary, where the RNA polymerase, that is the enzyme that is going to transcribe or synthesize the RNA, mRNA, and it is ending with the transcription termination site. This promoter and the transcription termination site, there are the regions like the transcription initiation site, then there is the leader sequence, then there is the translation initiation site, and between the translation initiation site and the translation termination site, we have the coding region, those are called the exons, and we have the non-coding region, those are called the introns.